Hello, my name is Soichi Ogata. I am a postdoctoral scientist in Dr. Kencho's lab at Department of Developmental and Cell Biology in University of California, Irvine. Today, I will show you the preparation of thin plastic section of Xenopus embryo. This procedure includes the bisection of late gastrous stage frog embryo, staining with antibody against the molecule of interest, infiltrate into the plastic, and make a 5-micron thin plastic section. This procedure can be utilized to study the immunofluorescence, immunohistochemistry, or in situ hybridization with very high resolution image. Okay, those are the small handy tools I use for embryo plastic sectioning of Xenopus embryo. This is a metal tweezer with dowel tip, and number one, this can be used to orient the embryo in plastic. And number two, this can be used together with this paintbrush to transfer the embryo from microtome to the mounting surface. This is a silicone rubber insulator which I put on the surface of the slide, and which makes a water pool where I can mount the slices of plastic sectioned embryos. Now I'm showing how to bisect the embryo, fixed embryo, for the assay like in situ hybridization or immunofluorescence detection. And to cut the embryo half, I use this very ultra-thin laser blade which you can buy in grocery store, I think.